we have a few different topics. We're going to get a little bit of everything today. So we're going to finish up carbohydrates. We'll move into a brief segment on a few of the organometallic reactions that you should know for the final. And then we'll wrap things up with some whatever we can get through with amino acids and proteins. So that's the plan. As far as scheduling goes, don't forget that you have your fourth and final midterm exam coming up on Wednesday this week, which will cover the enolate chemistry and carbohydrates primarily. And then whatever amount of protein we can get through today would also be fair game. But it will be a minority of the topics on the exam. So the bulk of the exam will be enolate chemistry and carbohydrates. Okay? And then you have your final exam coming up a week from today. So I will be posting my finals office hours. I plan on holding them every day leading up to your final exam, well, with the exception of the weekend. But Thursday, Friday, and Monday, I will hold office hours, and I'll let you know about that on campus.
Our final is at 10:15. Are you going to do it before?
Yeah, it'll be before. I think the way I've tentatively scheduled it, I have it written down somewhere. I need to go look at it again, but I'm tentatively scheduling it for the hour preceding your final. So, okay. Any other questions before we get started? All right, then let's go ahead. Let me start recording. Get going. Yeah. Oh, thank you. Oh, thank you. Okay, then let's go ahead and get started back into carbohydrates. Where we left off, we were talking about the glycosidic bond. The glycosidic bond, as we have discussed, is simply the acetal linkage that joins two monosaccharide units. All right, so you can see it shown on the slide here. We have two different units of glucose, and then the anomeric center of one is going to be bonded to another of the hydroxyls of the other. And just think about this in the context of regular functional group transformation. You've already talked about the chemistry of hemiacetals. And as you know, if you take a hemiacetal and add a second equivalent of alcohol to it, you make an acetal. And that is nothing different than what you're doing here. Our molecules are just a little bit bigger, okay? So what I'd like to do now is talk a little bit about how we classify these. We already discussed last time when we talk about the forming a disaccharide, one of them, because we're making an acetal, that means by definition, one of the two has to be providing the hemiacetal. Or said another way, one of the monosaccharides must be giving its anomeric center to the bond, right? And we call that one the glycosyl donor. And the other one that's providing the OH group to the acetal is the glycosyl acceptor. All right. So as you can see, you know, you can't just, when you're talking about a disaccharide linkage, I can't just say, oh, I have a glucose-glucose disaccharide, because that is very ambiguous, right? It's fine that you're putting together two glucose molecules, but how are you putting them together? Obviously, one of them is going to be giving its anomeric center to the bond, because in order to make an acetal, you have to have a hemiacetal. But in theory, any of these other OH groups could be adding to the bond. So you have to specify, it's not just a glucose-glucose disaccharide. What two carbons are being linked in that glucose-glucose disaccharide? So in this particular one, you can see it right here, I'm linking the C1, the anomeric center of the donor, as always. It is being linked to the 4 position of the acceptor. So I would say I have a glucose-glucose 1,4 disaccharide. Now that still is ambiguous because I have not addressed the stereochemistry at the linkage, right? Now when you're looking at stereochemistry, as always, we're looking at the acceptor. I'm sorry, rephrase. You're looking at the donor, right? It's always referencing the anomeric center. And so this glycosyl bond has the anomeric position of the donor in a downward direction. And so as we discussed last time, whenever you have the anomeric center, the oxygen going in a downward direction, we call that an alpha. So this would be an alpha 1,4 glycosidic bond. Okay? So let's do some practice with that. Here's a couple that are not too far of a stretch, I don't think. If we're looking at this glycosidic linkage, what 2 carbons are being linked there?
One and 4.
Yep, it's 1 and 4 again. And what's the stereochemistry?
Beta.
Yeah, it's beta. Now don't get confused. Some people look at that and they get a little bit mixed up. They say, well, I don't know, because it's going up with perspective to this monosaccharide, but it's going down from the perspective of that monosaccharide. You have to remember, the only thing you care about is the anomeric position. The only thing you care about is the perspective of the glycosyl donor, not the acceptor. Okay. What about the second disaccharide here? What 2 carbons are being linked in this?
That's a 1,3.
Yep, it's a 1,3. And the stereochemistry? It's beta. So that would be a beta 1,3. Okay. Let's do a couple that are a little bit different looking. Oh, I have this on there just for fun. This kind of shows you what they call the relative sweetness. And what you can see is that there's a bunch of kind of natural sugars there, and then there's some artificial sweeteners down at the bottom. And the thing that's kind of remarkable about this is, number one, a lot of sugars really aren't that sweet. If you consider sucrose, this is the one that we're probably most familiar with, has a sweetness, a relative sweetness of 100. Take a look at a lot of these other sugars. They're really not terribly sweet in comparison, okay? But then take a look at your artificial sweeteners. It's crazy. I don't really use a lot of artificial sweeteners, but I do like some sweetener in my coffee. I usually just use sugar, but you know, in a pinch, if there's no sugar around, I will reach for the pink packets or whatever they have. And I've ruined many a cup of coffee by adding the same amount of the artificial as I do regular sugar, just because I'm not used to it. And yeah, and that explains a lot. But anyway, that's neither here nor there. So let's go ahead and look at a couple more examples. How would we name this disaccharide? First, what are the 2 carbons being linked? Yep, 1 and 6. This one is also sometimes tricks people up because they sometimes forget to count the C5. Okay, but yes, that is a 1,6 linkage, and the stereochemistry would be
Alpha.
alpha 1,6. Now, this one I put on there because, well, mainly because it's the most common sugar that we all deal with on a daily basis, probably. This is sucrose. The reason it looks a little bit weird is because it involves glucose, which is normal. We've been looking at that the whole chapter long, but this one is a little bit odd looking, and the reason it's a little bit odd looking is because it is a ketose sugar. We haven't really looked at a ketose sugar yet. And so let me, cover my page here. So when you're looking at that ketose is actually fructose. The reason why these look a little strange is because the anomeric center is not the 1. The anomeric center is the 2 because that's where the carbonyl is. So when this cyclizes, it ends up doing this. The C1 is exocyclic here. The C2 is your anomeric position because that's where the carbonyl is. Okay? So, with that in mind, let's go ahead and see what the glycosidic bond between the 2 monosaccharides of sucrose is. First off, what 2 carbons are being linked here? Yep, that's it, 1 and 2. Second question, which one is the glycosyl donor? Remember, the glycosyl donor is the one that's giving its anomeric center to the bond. Yeah, this is a rather interesting case where both of them are technically glycosyl donors. So the rule is you have to give stereochemistry from the perspective of the donor. That means we have to give 2 stereochemistries here. So this would be what we call an alpha beta 1-2 linkage. Now, I'm not going to give you something like that on your exam. I just wanted to show it to you because, again, it's probably the most common sugar you deal with every day. But you should be able to name the simple glycosidic linkages, but I'm not going to give you one like that. Okay? Any questions on this before we move on?
What's the other slide that you have?
The handwritten one?
Yes.
Okay. And by the way, like I said earlier, this is fructose. Okay, then let's go ahead and we'll finish this chapter up looking at the polysaccharides. So we've spent a good bit of time looking at the characteristics of monosaccharides. We started looking at how these kind of start coming together to make the disaccharides. We'll wrap this up with the polysaccharides. When we talk about polysaccharides, these are simply polymers of monosaccharides. Generally, we say 10 or more. Okay, but in nature, these can be found in units in sizes of hundreds to thousands of monosaccharides long. Okay, very, very large macromolecules. Now, when we talk about the polysaccharides, they can come in two broad categories. What we call the homopolysaccharides, meaning that they're composed of only one type of monosaccharide unit, or we have the heteropolysaccharides, and these are composed of more than one type of monosaccharide unit. We're going to focus on the homopolysaccharides. They are by far the most common. So the first one we're going to look at is cellulose. Cellulose is the most abundant material on the planet, biological material on the planet, and it is the structural material found in plants. So this is a homopolysaccharide of glucose, and the kind of the distinguishing feature here is in the linkage. All right. Now, if you think about it, you know, one of the primary uses of glucose in animals is to be used for energy, right? For glycolysis. Now, in order to do that, in order to do glycolysis, you cannot just get an entire polysaccharide across a cell membrane. It's just too massive, okay? So in order to use the glucose from these polymers in glycolysis for energy production, you first have to break it up into pieces, into the individual monosaccharide unit. Right? This is the process of digestion. So in order to use cellulose as energy, we would have to be able to hydrolyze or enzymatically cleave that beta 1,4 linkage. And we don't have the enzymes to do that. Right? That's why if you get hungry after lecture, you can't go graze in the courtyard over there. Okay, you cannot eat grass because you cannot get energy from grass, right? Because you cannot digest this linkage. Now, that's not to say nothing can. I mean, plenty of animals graze all the time, right? Horses and ruminant animals like goats and whatever else, cows. They have those enzymes that can digest this linkage, and so they can get energy from that, okay? But we cannot. So let's go ahead and look at the next one. The next one is remarkably similar to cellulose, and this is starch. Starch is actually composed of two different polysaccharides. Starch is composed of amylose, which again is very, very similar to cellulose, and it's composed of a second one called amylopectin. Now the amylose, like I said, is almost exactly like cellulose, the only difference being in the linkage. So instead of being beta 1,4, it's alpha 1,4. And we can get energy from from starch because we can digest starch. We have these enzymes called amylase enzymes found in both our saliva and in the gut that can break that alpha 1,4 linkage. So we can, and probably most of us do, get a lot of our energy in the form of starch, right? From breads and pastas and potatoes and things like that, right? The amylopectin, the second polysaccharide in starch, is similar to amylose in that it's mostly an alpha 1,4 linked polysaccharide of glucose, but it's branched. And the reason it's branched is because 1 in every 30 monosaccharide units or so, you have an alpha 1,6 linkage. Okay? So those are the two polysaccharides that make up starch. And there's a few others. We're not going to get into all of the polysaccharides, but another one of the important ones that's another polysaccharide of glucose is that you probably are familiar with. It's called glycogen. Glycogen is the way animals store carbohydrates. So, right after you eat, there's an abundance of glucose in the blood, and the body stores in the liver, kind of tucks some away in the form of glycogen, so that during times where you're not eating, you know, say like a few hours after you've eaten something, when the blood glucose level drops, you can mobilize that glycogen. The body starts breaking the glycogen up and sending it into the bloodstream to help maintain a relatively constant concentration of glucose in the blood. So, but you don't have to worry about glycogen. We're not going to talk about it here. The last thing I wanted to mention involves the cell surface oligosaccharides. So for a long time, people thought carbohydrates were pretty boring. I know nobody here thinks that, but they thought it was just simply an energy production thing. You know, you eat sugary things or starchy things, the body breaks it down into glucose, and it goes through glycolysis into the story. It turns out that carbohydrates do so much more in physiology than just provide energy. And this was not really recognized until fairly recently. So I'd say between the 1980s and the 90s was when it really started to get going, and continues to today. And there's an entire field, they call it glycobiology, where people study what these carbohydrates, what these sugars actually do in the body. And the cell surface oligosaccharides are the best understood to date. And the most notable example are the blood group antigens. And so if you look at the blood group antigens, it's just an oligosaccharide. And what's really striking to me anyway is the subtlety. If you look at the 3 blood group antigens, they are all the same up to a point. They have the first 3 monosaccharide units that are identical to each other. N-acetylglucosamine, galactose, fucose. All of them share that. The only difference is in the terminal sugar residue. Group O lacks a 4th sugar residue. A has N-acetylgalactosamine, and B is galactose. That subtle difference in a single monosaccharide is the difference between the blood group antigens. And you think about the massive physiological difference that has. You know, if you tried transfusing a group A patient with group B blood, it's not going to go well for that person. And again, the difference is so subtle, just a single monosaccharide. And so people are beginning to realize that these monosaccharides, these carbohydrates in general, have a massive physiological role in virtually everything, ranging from the way cells communicate with each other, to inflammatory response, to the way cancers metastasize, to the very initial interaction between the sperm and the egg cell during fertilization. And also, importantly, how many bacteria and viruses enter the host cells. Many viruses and bacteria, that first interaction before they can invade a host cell is a carbohydrate-mediated interaction. And so there's been a huge interest in this in the past couple decades. That continues. And one of the reasons why this as a field is so late in developing is the complexity. Carbohydrate chemistry is hard. Believe me, I came out of carbohydrate lab. It is not trivial. And people before just a couple decades ago really didn't have the means to synthesize carbohydrates. You know, if you're trying to study the physiology of a particular compound, whether it's a peptide or protein or carbohydrate, you really have to be able to synthesize it to make enough of it to study, you know? And synthesizing something like this is not easy. The protecting group strategies alone is enough to drive a person crazy. But beyond that, let's say you can synthesize. Let's say you devised a way of synthesizing something like this. What do you think that would look like in the NMR? So even if you can come up with ways of synthesizing these oligosaccharides, trying to characterize them is another just mind-blowing exercise. I mean, it is extremely difficult to characterize these things. And you really require very advanced mass spectrometry, very advanced NMR techniques to do it, which just didn't exist more than a couple decades ago. So, and what I'll end with is this, it's an electron micrograph of a cell membrane. You can see this dark portion here, this is the interior, this is the cytoplasm of the cell. You got that dark black line, which is the cell membrane. And then you've got all this stuff out here. It looks like the thing's growing hair. That is carbohydrate. And so you can see just how literally covered our cells are with these things. And it's just now beginning to be understood what they're all there for. So it is a very active area of research in chemistry and biochemistry and basic biology as well. Okay. Okay, any questions on the monosaccharides or the carbohydrates? Okay, then if not, then we'll switch gears a little bit, and I want to look at some organometallic stuff with you. So, the first thing that I think I'll get into a little bit is, because this will lead us into the first of these reactions, of these organometallic reactions. The first one that we're going to look at is called the Simmons-Smith reaction. Before we can really get into the Simmons-Smith, we have to define something, and this is what is known as a carbene. A carbene is an electron-deficient, neutral carbon species. Okay. The way it was made originally, they used to make it in a couple of different ways. You can take bromoform and treat it with base, and it does a rather interesting reaction. It does an elimination, but not a normal elimination. When I say a normal elimination, that's what we call a beta elimination. And this is like your E1 and E2 style eliminations. Taking bromoform and treating it with a base will actually do what's called an alpha elimination. And what that means is that the hydrogen and the leaving group are actually on the same carbon. So what this does is the base will come in, grab the proton, and then cause a bromine to leave. And so what you end up with here is two bromines and a lone pair. So this is, number one, it's neutral. You know, you can calculate the formal charge of the carbon, and it's neutral. But it's electron deficient as well, right? If you look at that, carbon does not have its octet. And so these, as a result, are quite electrophilic, right? The carbon doesn't have its octet and it wants it. So it's going to find electrons wherever it can. Now, the other way that this used to be made is through diazonium chemistry. We've talked about that before as well. If you take diazomethane, and you treat it with heat or light, you can cause the nitrogen to leave. And again, you're left with this electron deficient carbon species. The problem with doing this is trying to heat or irradiate diazomethane was fairly explosive and pretty dangerous. So that's what Simmons-Smith really did. Through the use of their chemistry, they were able to come up with a way of generating these carbene structures without the risk of blowing themselves up. So that's where we're going to start this. And I'm going to switch over, I think, to some slides that I have on organometallics. So what Simmons and Smith did was they took diiodomethane, CH2I2, and they treated it with zinc. See that shown right here. And what that would do is they learned that it would react with alkenes. And it did that through this kind of a interesting, how they call it, a butterfly transition state. But in the end, what ends up happening is the CH2 will remain in place, will be bonded to the other two carbons of the alkene, and then you end up losing zinc iodide. So what Simmons-Smith effectively does is it takes alkenes and it converts them into cyclopropanes. And this was kind of important because we don't really have a way of making those except for the Simmons-Smith reaction. So let's go through and see if we can figure out what a couple of these would end up looking like. So we're starting, the first example deals with simple cyclohexene. If you do the Simmons-Smith reaction on cyclohexene, you simply will convert the double bond into a three-membered ring. So this would end up looking like that. Now there's some facial selectivity here, meaning that if you had something like the example below, you're going to get the three-membered ring adding to the least hindered side. So we've seen molecules kind of like this before. Think back to experiment 6 when we were doing the oxidation of borneol. We know that that top face of that molecule is quite hindered and kind of blocked off by the big methyl groups. And as a result, the product that is favored here is the three-membered ring towards the bottom. Something like that. All right.
What am I looking at?
You're just adding a CH2,
Okay.
so it just makes a three-membered ring. So now we have a tricyclic system. Okay, so the Simmons-Smith is nice and all. It's fairly limited in scope because, again, it's just making a three-membered ring, and how often do you need to do that? But it is something that you should be aware of because you may see it on your final. Okay, the next couple reactions are much, much more general and exceedingly powerful. In fact, they're so extensively used. The two people who developed them shared the Nobel Prize in 2010, I think. So it's fairly recent chemistry, and again, very, very powerful. The first of these is what's known as the Suzuki coupling, or the Suzuki reaction. And what Suzuki does is it takes an alkyl halide, so you can see it's typically bromine or iodine are used, and the other reacting piece is what's called a boronic acid or a boronic ester. And that just depends on what's attached to the boron. So it's always going to be some R group, some alkyl type thing. And it's either going to be an OR group, as I've shown here, in which case it's a boronic ester, or you can also see just OH groups used there, and that would be the boronic acid. Right? But it's going to use a palladium catalyst, and I just have L there. What that L stands for is just a ligand, and a ligand is just some group. It's usually some big carbon group. And so it doesn't really matter what that group is. There's all sorts of different ones. The important piece is the palladium. Right? And under basic conditions, you end up coupling the two R groups, the R group from the halide with the R group from the boron. Okay, so what that really looks like is something like this. So here's an example. We'll go through a couple of these and try to figure out the product in just a second, but I want to show you. So this is your halide. You can see it can be an sp2 carbon as well. It doesn't necessarily have to be an alkyl halide, but some sort of a halide. And then here's our boronic ester. And all we're doing is putting the carbon that is bonded to the boron, and we're putting that on the carbon that's bonded to the halide. And as you can see, stereochemistry is retained here. We started with the E double bond, and we make the E product. All right. You can see another example of it shown here. We have the aromatic iodide, and we have this boronic ester. But again, all we're doing is putting the carbon that's bonded to boron onto the carbon that's bonded to the halogen. All right. One of the reasons why this one and the next reaction are so powerful is because they make carbon-carbon bonds under very mild conditions, and they're incredibly versatile. You can make almost any kind of carbon-carbon bond you want, number one. Number two, you can do them in many, many different solvents, including water oftentimes. Let's go ahead and see if we can figure out a couple of these that don't have the product written for you. All right, so you can see in the case, the third reaction here, we're looking at an aromatic halide and an aromatic boronic ester. Nothing's going to change. We're still putting the carbon bonded to the halide, and we're going to bond it to the carbon bonded to the boron. And so in this case, we would simply make biphenyl. All right, any questions on this? Okay, there's a couple other examples that you can work on, see if you can predict the products of on your own if you like. But I want to go and look at the next reaction. The next one is another organometallic reaction, and it's quite similar to the Suzuki coupling. This one is known as the Heck coupling. The main difference here is that instead of using a boron compound, it takes the halide and it simply couples it to an alkene. The only necessary requirement here is that the alkene must have at least one H. Okay, so it can't be a tetra-substituted alkene, in other words. So some would argue that the Heck is even more useful than the Suzuki coupling for a couple reasons. Number one, it is even milder conditions than the Suzuki. So instead of using a hydroxide base, you're just using a very mild, usually triethylamine, just an amine type base, which is a lot weaker than the hydroxide. So it's more appropriate in case you have base-sensitive functional groups. The other thing that's nice about it is it uses a halide and an alkene. There's no need for a special boron reagent. Not that that's that much of a hindrance anymore, because they have literally catalogs of boron reagents that you can buy specifically to do Suzuki couplings. So they are very easy to come by, but you know, alkenes are a lot more abundant and easier to make. So, but in the end, what you're going to do is you're just going to couple the carbon that bears the halide to the carbon of the double bond. Okay. So in a case like this, you can see that we would end up making just styrene. All right, let's see if we can figure out maybe a couple of these that don't have the products. So in this, let's look at this one. So we can see that it's going to be that carbon that has the halide, and it's going to be bonded to the carbon that has the vinylic H. So in this example, we would make and here again, you can see that stereochemistry is retained. You can do one more. Let's look at the next one. So in this case, we would take the carbon that has the halide, bond it to the vinylic H. All right. And this is modern chemistry. You would be hard pressed to find a synthesis in a journal these days that did not use organometallic reactions in the synthesis somewhere. It is a very, very powerful way of making carbon-carbon bonds, and they're used all the time. All right. Any questions on the organometallics before we move on? Okay, there's another one on here. If we had more time, I would cover alkene metathesis with you. It's another very powerful reaction. A person who developed that also won a Nobel Prize. But I don't think you're gonna see it on your final, so I think we'll just move into the proteins at this point. All right. All right, so we'll spend the rest of the next 30 minutes or so doing a crash course in amino acid chemistry. So, and we're gonna kind of approach this in the same way that we approach the carbohydrates. So the proteins are also biopolymers, meaning they are long molecules that are made up of a repeating monomer unit. The monomer unit here are going to be the amino acids. And so we will kind of focus a little bit first on the characteristics of amino acids, and then we'll ultimately build up to the polymeric proteins. All right? So when we talk about amino acids, they all have this kind of the same generic structure. meaning they have a carboxylic acid piece and they have an amine piece, hence the name amino acid. What distinguishes one from the others is what is at the alpha carbon. So the alpha carbon is that carbon that's next to the carbonyl, and it has on it what we call an R group, or you'll hear it called a side chain, and it is the nature of that R group that distinguishes one amino acid from the other 19. Okay? So just to give you some examples, we can break these amino acids. There are 20 that are naturally occurring. And we kind of break them up into different categories based on the nature of their R group. So we have what we call the nonpolar amino acids. And these are going to primarily be alkyl type side chains. And there are about 10 of those. We have the polar acidic amino acids, and these are going to be where your side chain has a carboxylic acid group. So there are 2 of these. You have the polar basic amino acids. These are going to be side chains that contain some sort of an amine functionality. There are 3 of these. And then you have the polar neutral amino acids, and these are going to be things where you have functional groups like alcohols or phenols or amides or something like that. And there are, do the math, I guess that makes 5 of those. All right. So I'm going to show you just some of the examples. You don't need to memorize the amino acids for this class. You'll get that fun when you take biochemistry. But just to kind of illustrate, just to orient you here, what you're looking at is, that's the backbone, right? You can see that each of these has a carboxylate alpha carbon amine, okay? All of them have that. And then you can see that the only thing that's changing is what's sticking down below the alpha carbon. So for glycine, glycine is the simplest of the 20 amino acids. It has just an H. As we go down, you can see alanine, you have the methyl, you have an isopropyl, you have the isobutyl, a sec-butyl, you have this benzyl group here, you have what's called an indole ring. So just a bunch of kind of non-polar hydrocarbon stuff as side chains there. For the remainder, you can see at the top, you have the 2 polar acidic, right? You can see that you've got this carboxylate group there, you've got a carboxylate group there. Here you've got 3 polar basic. You can see you've got an amine group here. You have this amine type functionality here. It's called a guanidinium group. And then you have this nitrogen ring here called an imidazole. And then over here you've got the 5 polar neutral amino acids where you can see alcohols and phenols and amides. Okay? All right, so the most notable thing about the amino acids is that because they have 2 ionizable functional groups, namely the carboxylic acid and the amine, at physiological pH, they are both charged. Right? They exist as doubly charged species. Then we call that a Zwitterion. Zwitterion is German for hermaphrodite, but anyway, they exist as this doubly charged species. However, they can change depending on the pH that they're placed in. So if you have a very, very acidic environment, everything will be protonated. So you can see that shown here. So the carboxylic acid is now protonated in neutral, and the amine is protonated in positive. If you take it to the opposite extreme, under very, very basic conditions, everything is deprotonated. The carboxylate is charged, and the amine is uncharged and neutral. It is only in that physiological range where both of them are charged and give you that zwitterionic form. One of the big characteristics that you often talk about when you talk about amino acids is what's called their isoelectric point. The isoelectric point is the pH at which the Zwitterion exists or forms. And for the neutral amino acids, that tends to be just below neutral, so in the 5 to 6 range. For the polar acidic amino acids, the isoelectric point is quite acidic in the 2 to 3, and for the polar basic, the isoelectric point is in the 9 to 10 range. And I want to show you why that is. So let's just take, we'll do with an acidic amino acid first. Let's show you aspartic acid. So I have drawn aspartic acid as it would look under very acidic conditions. Okay, everything's protonated. Now what we're going to do here is we're going to start imagining kind of titrating in base. So if we were to take aspartic acid and start, before I do that, let me give you the pKa values for this guy. The backbone carboxylic acid is about 1.88. Side chain carboxylic acid is about 3.65. And then the amine, the ammonium, is about 9.6. So if you start adding base to this, what's going to go first? What proton are you going to lose first? Or said another way, what is the most acidic proton? Yeah, which one?
The main one.
Yeah, the 1.88, right? That's the lowest pKa, that's the most acidic group. So as we start adding base, that is going to be the first to go. So let's go ahead and draw what this would look like without that proton. So at one equivalent of base, you'd have that. So the question is that a Zwitterion? Zwitterion should have one positive and one negative.
So yeah.
Yeah, that's your Zwitterion. So you have to think, okay, well, what pH would that be? Well, the pKa of that proton is 1.88. If you remember, if the pH is equal to pKa, you would have equal amounts of protonated and deprotonated. So at pH 1.88, you would have equal amounts of these. To get all of that, I'm going to have to increase the pH a little bit beyond this pKa. But not too much, because I don't want to start pulling that one off. So that's why with these acidic amino acids, the isoelectric point is somewhere between 2 and 3. It's kind of that sweet spot between those two pKa values. Okay? Let's look at the other extreme. Let's look at a basic amino acid. So this is the amino acid lysine. pKa values of this are 2.18 for the acid. 8.95 for the backbone amine. And then 10.5 for the side chain. So this is what lysine would look like under strongly acidic conditions. We begin to titrate base. The most acidic proton here is obviously the carboxylic acid, so that would go first. is that Zwitter ion? Remember, the Zwitter ion is 1 positive and 1 negative. So that is not it, right? Because I've got 2 positives and 1 negative. So I need to add more base. After another equivalent of base. Nope, that one would be deprotonated. How about now? Yep, that's 1 positive and 1 negative, so that would be our Zwitter ion. So the question is, at what pH does that happen? Well, go back to your pKa's. So at a pH of 8.95, you would have equal amounts of this guy protonated and deprotonated, so you need to go a little bit more basic than that, but not too basic, because we don't want to pull that one off. So somewhere between 9 and 10 would be the sweet spot where we would find our Zwitter ion for a basic amino acid. Okay. Now it turns out we can tell a lot of this information. We don't have to go through this exercise every time. You can get a lot of this information simply by looking at one of these. Remember these things? The good old titration curve. Probably hope you'd never have to see one of those again. But it turns out that we can get a lot of information about these amino acids and their isoelectric points and their pKa values simply by looking at these. Okay? So, one of the things that we can do with this is we can tell where the isoelectric point is. Because this, well, granted, this is a looking at glycine. So this is just one of your neutral amino acids. All right. So we can tell what the isoelectric point is simply by looking at what pH, what is the pH after 1 equivalent of base. So what you're looking at here, we've got the pH on the Y, you've got equivalents of hydroxide on the X. If we come over to 1 equivalent of base, that's where your isoelectric point is. Okay. Now, you can also tell where your pKa values are, because here, you might remember an expression. Does that look familiar? Anybody remember the name of that one? It starts with an H. The old Henderson-Hasselbalch. So we can use the, I'm going to illustrate a point here, hopefully. So if you imagine titrating in base, we're starting with everything protonated. After 1 equivalent of base, you deprotonate the carboxylic acid, and that makes your Zwitter ion. Now, what if we didn't add a full equivalent? What if we only added half an equivalent? Well, if you've only added half an equivalent, you've only done half the job. So we would have half protonated and half deprotonated. We'll come back to the Henderson-Hasselbalch. What happens in this expression up here if you have equal amounts. If you have equal amounts, that fraction goes to 1. And if memory serves, if you take the log of 1, that whole thing goes to 0. So at half an equivalent, what we're saying is that the pH is equal to the pKa. So that's another really important piece of information that this titration curves tells you that you don't even have to think about. I know that at half an equivalent of base, that's my pKa value. I know at 1 equivalent of base, that's my isoelectric point. I know at the 1.5 equivalent of base, that's going to be your second pKa. That's the pKa of the amine. So these titration curves do give you a lot of information, almost just, you don't even have to think about it, just look at the curve, and as long as you can interpret it, then you get all this information just looking at the curve. You don't have to do any calculations, you don't have to even really think about it. All right. We can use this differential protonation to our advantage. And some of you have probably done it before through a process called electrophoresis. So this is probably the most common way of separating out all sorts of biological molecules, amino acids, peptides, proteins, nucleic acids. But what you do, it's based on difference in charge. So for instance, if you had, this is a very simplistic example, but let's say you had 3 different amino acids, alanine, lysine, and aspartic acid. At a pH of 6, those would be differentially charged. So the alanine here would have a net 0 charge, the lysine a +1, and the aspartic acid a -1. So if you were to inject this mixture into a gel and apply a current, because they have different charges, they would migrate differently in the gel. And they would ultimately separate out from one another. So for instance, the mixture would be kind of shown here as in gray, and then you have the lysine with a plus charge, which would migrate to the negative electrode. The aspartic acid, which with its minus charge, would migrate to the positive electrode, and then the alanine with a net 0 charge wouldn't move at all. Okay? So this is a very, very common, if you take a biochem lab or a molecular bio lab, you will certainly be running gels all the time. Okay, so let's go ahead and talk about a little bit about stereochemistry. So much like the monosaccharides, amino acids, as you can tell, probably by looking, they also have stereochemistry, right? Because that alpha carbon for at least 19 of the 20 amino acids is a chiral center, okay? Meaning that if you put these two mirror images next to each other, they would be non-superimposable mirror images or enantiomers. And we're going to use that same terminology that we introduced for carbohydrates, we're going to use it for amino acids, namely that D and L. So when we talked about D and L sugars, we were interested in the orientation of the hydroxyl group. For the amino acids, we're interested in the amine group. So if the amine is pointing to the right, we call that D, and if it's pointing to the left, we call it L. So these two shown on the slide would be D and L alanine, and they are enantiomers. Now, for whatever reason, back in evolutionary history, nature chose to deal in one of these, and nature chose to deal with the L. So all of the amino acids that make up pretty much the biological world around us are L. There are a few D amino acids found in nature, but they are found primarily in bacterial secondary metabolites. I have a couple slides on amino acid synthesis. You don't need to know that. realistically, if you ever wanted an amino acid, you would find it in a catalog and buy it. Nobody really synthesizes amino acids anymore. So you can forget about those. Let's talk about how these come together. In the same way that we talked about the monosaccharides, disaccharides, polysaccharides back with sugars, we can do the same kind of thing with amino acids. We call them peptides. So if you have two amino acids coming together, we call that a dipeptide or a tripeptide or a polypeptide, okay, just depending on how many amino acids are linked. The way these become linked is the amine of one bonds to the carboxylic acid of the other, right? And as a result, what you end up making is technically an amide bond with the loss of water. And biologists refer to this specific amide linkage between amino acids as a peptide bond. All right. Now, when we start putting these together, what you can see is that these growing peptide chains have directionality. Right? There's always what we call a C terminus and an N terminus. Right? And whenever you're talking about a specific peptide, you read in that order. In much the same way, whenever you're reading a sentence, just a page, you always read left to right, unless you're Japanese, I think. They read the other way. But anyway, the peptides are all read from N to C. Right? So for instance, we would read this just by simply reading out the listing of amino acids. So it would be arginine, proline, glycine, phenylalanine, serine, proline, phenylalanine, arginine. Okay? Reading out the individual amino acids N to C. Right? Okay. So to do synthesis of these, you could imagine it might not be such a great idea if you wanted to make this dipeptide. So we have glycine and we have phenylalanine, and that's the dipeptide we want. Would it be a good idea for us to take these two amino acids and put in one of those coupling agents? Would we get the dipeptide we're trying to make this way? Yeah, you would make some glycine phenylalanine, but you'd also make a whole bunch of other things, right? You would make some gly-phe, but then you'd also make some phe-gly, which are not the same thing because there's directionality of these peptides. You would also make some gly-gly, and you'd make some phe-phe. So you only want to have one amine and one carboxylic acid exposed. And so because of that, we have to protect. And some of the more common ways we have of protecting, the carboxylic acid is easy. Generally, we'll just protect that as an ester. So a methyl ester there. The amine, we're going to protect like this, which is what we call Bach. So that when we do the coupling, there's only one way this thing can combine. You have one carboxylic acid, one amine, and that is going to make one amide linkage. Now the other thing that we have to make sure of is that the protecting groups are orthogonal, meaning we don't want to be able to deprotect both of these under the same conditions. Because what that will allow us to do is it will allow us to selectively deprotect, and then we can grow out the chain in either direction. Right? So using this type of protecting group strategy, I can remove the ester in base and grow out the chain that way. Or I can remove the Bach group in acid. And grow the chain that way. So it allows us to do whatever it is we want to do. And that's how this synthesis is oftentimes done. Now, it becomes quite tedious to do it this way, though, if you try to make like a decapeptide or 12, 15, 20, something like that, because you have to purify and characterize after every step. And if you have this iterative step of growing out the chain, one amino acid by one, by one, by one, that's a whole lot of purification and characterization. And it would take you years to make a decent sized peptide. So what has largely supplanted solution phase peptide synthesis is solid phase peptide synthesis. And this is really cool. In fact, the guy who introduced this back in the 70s got the Nobel Prize for this. But it uses these little beads, and the beads are functionalized in some way, and it grows the peptide on these little beads. If you've ever, I don't know, if you eat sushi, you know the little, they're called fish roe, the little eggs, the fish eggs, the orange things that they put on top of the rolls? That's what these beads look like. So, but the way it works, it's rather ingenious, is, like I said, you take the bead and it's functionalized with something, an amine, a carboxylic acid, something. And what you do first is then you put on your first amino acid. So you couple it to the bead. And this nitrogen here has a Y group. That's just saying it's protected in some way. So then you take that and you deprotect it. So now we have the NH2, and then you can put the next amino acid on. And the advantage of doing this is that you can run these reactions in these little things that look like syringes. They call them solid phase reactors. And then you have all your beads in there. You can add solvent, plus, say, the next amino acid, plus the coupling agent. And you just let them sit for a period of 30 minutes or so. And then you come back, and then you just drain everything out. So the peptide would be contained on the bead. It would be covalently linked to the bead. And then you can just rinse all of the beads with some solvent, let that drain out, fill it up with solvent again, and move on to the next step. So it dramatically shortens the length of your synthesis, and it also makes it much, much more convenient. So you grow out the bead as long as you grow out the peptide chain on the bead as long as you want, and then in the end, you cleave it off of the bead and collect it. So this is the way that peptide synthesis is done these days. They've actually even automated it. So it's a very convenient way of doing this. I think I'll stop there. I don't really want to get into the peptide analysis and I don't really want to get into the protein structure either because we only have 4 minutes left. So I think we'll stop there, and yeah, that is what your exam will go up through on Wednesday.